This video is for Mendelian Genetics Part 1. So just a few concepts to review concerning gene expression. As we begin talking about Mendelian genetics, we're going to talk about a lot of gene expression and what that means. Genes are just DNA segments, and they're inherited from your parents, and they are the blueprint for making proteins, and proteins in turn are what cause you to have the traits that you have. Blue eyes, black hair, tall, short, whatever it is. Here's a picture of Gregor Mendel. Gregor Mendel was a monk, and he was considered to be the father of modern genetics, or not modern genetics, more so just genetics uh, as a whole. And it's what we get our Mendelian genetics from. He did a lot of tests with pea plants and with bees, and from those tests, he was able to derive some of the most basic things that we know about genetics today. A couple of terms that we need to be familiar with as we're talking about Mendelian genetics. Two of them are character and trait. A character is something that you can inherit, like a physical feature, like eye color, hair color, height, width, all of those different kinds of things. Whereas a trait is a variation of that character. You have blue eyes, brown eyes. This cat has yellow eyes and weird ears. And so ear shape is the character. Weird ears is one of the traits of that character. A trait or a character can have lots of different traits associated with it. It's not just two, it could be several. Now, another term that we're going to use quite a bit is the term allele, and this is just refers to different versions of a gene. If you remember with chromosomes, most chromosomes in eukaryotic organisms have a homologous pair, and so each of those homologous chromosomes contains the same genes, but they might contain different versions of that gene. And so each trait then will have at least two alleles, one from each parent. One of those alleles can be dominant or recessive. A dominant allele is always expressed. It at least shows itself, and recessive traits are only expressed if there is no dominant trait available. And so normally take this picture here, allele for purple flowers versus white. We're going to say that purple is dominant, and it's always written with a capital letter. Recessive, always with a lowercase letter. You should remember this from the many hundreds of times that you've had this. And again, you get one from each parent. Each sex cell contains a full copy of alleles, but just one version of that. So you can pass on, some, like, for instance, if you have blue eyes, blue eyes are recessive, and so you can only pass on the trait for blue eyes. And so more than likely, unless there's something wrong, if you have children with someone with blue eyes, then you're probably only going to have blue-eyed children. So a couple of other terms to be familiar with. One of them is true breed versus hybrid. A true breed or a pure breed, these are organisms that only have one type of allele for a trait, meaning that they are what we call homozygous. Homozygous. Now in the past, when I've done this, for some reason this is where I start to lose people. Homo means same. Zygous means like a zygote. And so homozygous or true breeds are either going to be all dominant or all recessive. And so that means they can only pass on one kind of trait. Whereas a hybrid, I don't think I have another picture. A hybrid is a mix. Hybridization is the process of creating an organism with two different kinds of alleles, or a mix, and hybrids then are referred to as heterozygous, or like big A, little a, has one of both alleles. They still express the dominant trait, but they have one of each allele. They're able to pass on either the dominant or the recessive trait. Now, phenotype versus genotype. This is another thing that people will oftentimes get mixed up. Pheno literally means physical. It's nice and easy because physical starts with pH. Pheno, pH. And the phenotype is the physical trait. This is what you can see. Blue A or blue eyes. Type A blood. Tall. Brown hair. Weird ears. Whatever. This is what you can see. This is the phenotype. It has nothing to do with the letters or the genotype. It has nothing to do with that. It's just what you see. Whereas the genotype is, geno means genetic. So this is like the actual allele type that you have. A genotype can be homozygous recessive. Homozygous recessive. Whereas a phenotype would be like, a genotype could be heterozygous or homozygous dominant. Notice there's no heterozygous dominant or recessive because it's both. And so again, this is a fairly complicated process for some, and so just make sure you don't get these two mixed up. Now there is such a thing called a test cross, and a test cross is just to find out what the genotype of an organism is, and the best way to do that is to cross them with a homozygous recessive. Why is that? Well, if you cross them with a homozygous recessive, then this is going to allow you to see whether or not the dominant trait will be expressed. So homozygous recessive here, little p, and if you cross it with the big P, little p, then you'd expect occasionally for these two little p's to mix up and you'd see some white flowers. Whereas if it was big P, you would only ever see purple flowers as the offspring. And so it's a way to determine genotype. Now everyone's favorite thing is Punnett squares, and Punnett squares are just charts and they show possible genotype outcomes. You've been doing these since 6th grade, you know what they are. Monohybrid, dihybrid, trihybrid crosses are usually ones that are used with Punnett squares. The monohybrid cross is just one trait. So it's one set, you know, big P, big A times little a, or whatever. And so you only need 4 squares for that at most. If you had something like this, you'd really only need 1 square for it because they're all the same. You could kind of do like that or whatever. But the point is that 1 trait, 4 squares. When you get into 2 traits, it gets a little bit more difficult because then you just have to start figuring out different allele combinations and gamete combinations. So if you have something that looks like this, big A, big B, little b, in order to draw your square, you have to figure out how many different combinations you can get with A and B, like big A, big B, big A, little b, little a, big B, little a, little b. Well, that's all well and good. We're going to learn a different way, and I'll show you how to do that when we're doing crosses. And you're actually going to do an exercise on this today. And so let's say you have a cross like this. Normally a cross like this would take you 16 squares to complete. you'd have to do out all the different combinations and so forth, and you'd have this big giant thing, and you'd have to fill it all in, yada yada yada. No one wants to do that. And so there's a way to do this just by using probability, where I could ask you, for instance, tell me what percentage of organisms will have this genotype, and you do not have to do the cross. Well, how do you do that? You take each one in turn. So let's first consider the A's. What is the probability that if we cross those A's, we'll get this outcome? Well, we can do it out if we need to here. We shouldn't have to do that. 1/2. Well, let's look at the B's. What's the probability then if we cross this with this, that we'll get this? 1/4. So 1/8 of the time, those two organisms are going to have that combination. And so now you have something like this. That's going to require, I don't know, 256 squares or something to solve for something like this, which would take you like an hour to do, and lots and lots of frustration. If I told you to tell me how many organisms had that genotype, you should be able to solve for this fairly quickly. Well, the A is going to have 1/2, the B is going to have 1/2, the C is going to have 1/4, and the D is going to have 1/4. 2 times 2, 4 times 4, 16 times 4, 1 out of 64 organisms born to that combination here will have this genotype. You can do that with 10, you can do that with 40, it doesn't matter. So it's nice and easy doing it with the probability method as opposed to writing out all the tricky little squares. So moving forward, a couple of things that you need to know concerning Mendel. His law of segregation. His law of segregation. Segregation means to separate. This has to do with the homologous chromosomes, or the sister chromatids, separating from one another in meiosis. We talked a little bit about this on Friday when we talked about meiosis. And so this is what's happening in anaphase 1 and anaphase 2 as they are separating. And let's look at this as far as the letters are concerned. And so, well, actually, let's just draw a cell. Here's a cell. Remember, it's got one homologous pair. And let's just say that the allele combination here is big A, little a. Well, as these, so this DNA is going to duplicate, and then there's going to be 2 divisions, right? 2 divisions. Over here you've got big A's, over here you've got the little A's. And so what the law of segregation says is that each one of those alleles, the big A's and the little A's, separate from one another so that each one has an equal chance to be combined with the mate of the opposite side. So that little a could combine with a little a from another organism and create a homozygous recessive individual, even though this individual started with a heterozygous. And so that is the law of segregation. Segregation happens during these steps, during anaphase. So make sure you don't get that mixed up with independent assortment. Now, what is independent assortment? This basically states that there are different sets of chromosomes. Remember, we talked about on Friday how the crossing over creates 4 genetically different organisms, or 4 genetically different cells. Well, independent assortment has a lot to do with that. And so this has more to do with So you have something like this. Well, independent assortment says that A's, B's, and C's all independently assort themselves so that you could have different combinations of the three, like this, or or that, and so forth. Lots of combinations because they all independently mix. It's completely random. The big ones don't stay together. The small ones don't stay together. They mix. Now, this isn't 100% correct, but this is important, and this laid the groundwork for later study. But we know some different things because of linked genes, which we'll talk about later, crossing over, and some of the other different things. Now, one thing to understand concerning these Punnett squares and concerning probability as we talk about genetics, probability has no memory, or chance has no memory, meaning that the likelihood of something happening doesn't change just because it's happened a certain amount of times, in a certain way. So, for instance, my wife and I have 3 girls. Well, the probability of us having a son, if we were to have a 4th child, is still 50-50. That doesn't change just because we have 3 girls already. You flip a coin, the probability of getting heads is 50-50, regardless if you flip the coin 900 times and you've gotten tails previous, which would be kind of outlandish, or there's probably something wrong with your penny. But probability, again, it's just a fraction, and you guys, a lot of you guys who have had stats, you understand this, has no memory. It doesn't remember. It's always the same ratio, unless, of course, there's something that is toying with the probability, and we've messed with some of that when we've done our chi-square analysis. So, last little thing there in your notes, quick way to assess probability in a mating cross, and I'm going to give you a sheet to help you understand this, and this is that rule of multiplication that we were talking about back here. This exercise I'm going to give you is pretty straightforward. I would like for you to attempt it on your own so that you can understand it. You can rest assured that you'll probably see a problem like this on a test coming to you soon. The AP exam has had questions like this the last couple of years where you're given like 5 or 6 different gene combinations and you're asked to give the probability of them. At the end of the day, it's just multiplying fractions, but if you don't know how to do it, it's an easy question that you should have gotten right, but now you've got wrong. So hopefully practice like the one I'm giving today will help you with that.